All right, let's go and get started. We've got the blood and heart this week from pages 42 to 46 in your lab manual. And as always, any questions that you have, please feel free to email me. This lab coincides with a lab activity that I have on the Blackboard site page for Blood and Heart under the lab content. So the first 3 pages of that activity will go along with this PowerPoint, and then the last 2 pages will be tidbits from this PowerPoint and presentation so that you can know a little bit of what you need to know for the lab quiz. But it doesn't necessarily go in order in those last 2 pages. Those are just tidbits that you need to make sure you know. And I'll present on all that in this particular presentation. Also, what you need to know, the models slides, or I guess models item in your lab is going to be coinciding for this week and next week, but use it for this week. Some students have been finding issues finding the model slides. I know I didn't have it on there for one of them and I extended it because of that, but regardless, you should be able to find the models on there for this week's lab. So let's get started. So starting off with objective 1, you need to be able to determine what is blood. So 55% of blood is made of plasma, which is going to be like water, proteins, nutrients, and hormones. So like albumins and globulins, hormones will travel in the plasma, and nutrients. This is the matrix of the connective tissue that is blood. So it's not living. The formed elements make up 45% of blood. So these numbers aren't necessarily going to be consistent from where else you'll find them. Sometimes you'll find 44% and 56%, or but what we're going with is 45% and 55% here. The 45% of formed elements in your blood, that is all the living constituent of blood. So you have your erythrocytes, which is your red blood cells. This will carry oxygen. You have your leukocytes, about 1%. This will carry things that will help fight off infection. We'll determine what types of immune cells are going to be involved with the different immune system functions when we get to that section. But for right now, just know that about 1% is going to be leukocytes. And inside that 1% also is some platelets. So these are little fragments of cells that will help with clotting. So they are the smallest blood cells, and they will help form clots. So that's one of the little tidbits on your activity sheet. Thrombocytes, they are little platelets that help with clots, smallest blood cells. All right, so the leukocytes and thrombocytes make up what's known as the buffy coat here. It's approximately 1%. 45% is red blood cells. I guess it's about 44%, and then about 1% is the leukocytes, and then 55% is the plasma. All right. Next we can talk about, next we can talk about the blood cells. So this is objective 2. I think I have it reversed here, but it's okay. We'll just go ahead and do it in this order. The objective is to basically be able to identify blood cells and leukocytes. So the red blood cells are pretty straightforward. They just look like little donut discs, little erythrocytes. They're pretty straightforward. You can see them all over the place here. In each one of these pictures, we see red blood cells. All right, these are all red, and they're blood cells. So they're not going to have nuclei. They're anucleated. So they don't actually undergo mitosis. So when they die, they get recycled in the blood, or via like the liver and the pancreas and the spleen. They get recycled. But regardless, they don't have nuclei. So that's one way to tell if it is a red blood cell. And we know if it's a nuclei because it's a dark purple stain. So this stain is staining for DNA. And so in all of these, all the DNA is being stained purple. And so that shows us that all these white blood cells have nuclei. And the way that we can see these nuclei is how we can determine what white blood cell we're looking at. Okay, so we have basophils, eosinophils, neutrophils, monocytes, and lymphocytes. And we can determine what we're looking at based on the way that their nuclei look. So with basophils, you can't really see a nuclei. It just looks like a bunch of dots all over the place, this chaos. So you can pretty much not see the nucleus at all. So if you can't really distinguish it, you're looking at a basophil. if you can see two lobes of the nucleus, two distinct regions, and it's kind of like this reddish orangish color, right? You look at the other ones, there's no reddish orangish or like burgundy pinkish going on here. This deep pink. This stain is staining for the eosinophil. You can distinctly see two nuclear lobes, two nuclei. My mouse disappeared. Two nuclei. Okay, kind of looks like this one's stringing off a little bit, but this is 1 and 2 nuclei. Eosinophil, 2 nuclei. And then in neutrophil, we're going to see multilobular nuclei. So what I mean by that is 3 or more. So there's 2 here in the eosinophil. The neutrophil, it looks like there's multiple lobes, right? 3 or 4 or 5, 6 lobes here maybe even. That's how we know we're looking at a neutrophil. So this lightish pinkish background stain, lots of lobes, bunch of balls, bunch of nuclear balls or lobes kind of sitting in here. Neutrophil. And then we have our monocytes. Our monocytes kind of look like a horseshoe-shaped nucleus. Kind of looks like a Pac-Man. So this monocyte has 1 nucleus, and it's looked like a little Pac-Man horseshoe here. And it's got this purple background too. And then a lymphocyte kind of just looks like a full moon. So the way the lymphocyte and the basophil get commonly mistaken. The lymphocyte has a bluish, purplish background. The basophil has some pink back there. The lymphocyte has 1 big globular nucleus, a full moon nucleus. Whereas the basophil has a bunch of polka dots. All right, so we're kind of looking at a really misshapen nucleus in the basophil and a very solid nucleus in the lymphocyte. Kind of difficult to determine all these, but when you're looking at the white blood cells, what can you distinguish from their nuclei? That's how you distinguish these white blood cells. We don't need to know the functions for them, as this is just the anatomy portion of the class. The functions will be covered in lecture. Now we can move on to objective 3, looking at some of the microscope stuff for this lab. This is heart tissue, all right, cardiac muscle tissue. So what we're looking at is striated tissue. These are these long striations. These are the different myosin and actin proteins overlapping each other that we talked about last week for muscles. And so these intercalated discs are going to be separating or kind of sporadically separating different cells from each other. So this is all one cell here. And these intercalated discs are separating the cells. And this allows smooth connectivity of all the cells simultaneously for full unit contractions. So it's helping with the electroconductivity of the heart muscle contracting, so that we constantly are contracting it at a smooth rhythmic pace rather than something like a bicep muscle that doesn't necessarily need to be contracting on a regular scale. So this heart tissue is making up this heart tissue is made up of cardiac muscle tissue. These cardiac muscle fiber cells are made up of intercalated discs separating each other. Okay, so it's pretty similar to skeletal muscle, besides we have intercalated discs. And also the nuclei are a little bit less abundant. There's a lot more nuclei in skeletal muscle. Now we can look at objective 4, which is going to be exploring the different external anatomy of the heart. So first I want to draw your attention to this, look at this bluish sac that is surrounding the heart here. This is called the pericardial cavity. So peri meaning around, cardial meaning heart. This is just the cavity that surrounds the heart. Okay, so this blue is this fluid that's going to be in here. There's a cavity that's around here. The lining right on the inside of that cavity, lining the heart, is called the visceral pericardium. Visceral meaning organ, so it's the pericardium, the heart, surrounding heart layer around the organ. So we're talking about the heart, it's surrounding the heart. Pericardium surrounding the heart, visceral meaning it's surrounding the organ itself. The parietal pericardium on the outside of the pericardial cavity, this one is going to be lining the area that the heart is placed in, the cavity that the heart is placed in. So the pericardial or the parietal pericardium is going to be on the outside of this pericardial cavity. So lining the heart directly is the visceral pericardium. Lining the outside of the cavity that's connecting it to the walls, like the thoracic cavity on the outside, is the pericardial, or the parietal pericardium. All right, we're going to come back to this idea when we start talking about the lungs too. We'll have visceral and parietal pleura that are going to be lining the lungs. Then we have this distinguishing of where we're looking at the heart between the apex and the base. So the base and the apex aren't where you think they might be. The base, when we think about a base, is being at the bottom. Well, in our case, it's where the blood enters the heart. So the base is at the top. The blood vessels, the major blood vessels entering and leaving the heart are at the top of the heart. That's the base. All right, so up here is the base. So what we're looking at here for this base is going to be kind of right around, which we'll identify here shortly, the right auricle. So pretty much where the inferior and superior vena cava are going to be entering into the heart. Okay. And then we have this apex. The apex is not very important. It's mostly just the point of the heart. It's just an area that we can look at, and when we're looking at a heart, maybe we're holding it and we're trying to identify what we're looking at, maybe we can find the apex and then go from there. So it's just kind of where everything comes to the point here of the heart. Then we have this anterior interventricular sulcus. All right, so when we're looking at our hearts in lab, obviously this video is for people that aren't in lab, this anterior interventricular sulcus is running diagonally in front of the heart. So we're looking at an anterior front side view of the heart. This anterior interventricular sulcus is basically interventricular, which means it's between interventricles, the left and right ventricle. And it's a sulcus. It's this area, this vasculature running veins and arteries in between the ventricles to supply them with the nutrients that they need. And it's anterior because it's on the front side. So we know we're looking at the front side of the heart because we're looking at this anterior interventricular sulcus. And then we have the coronary arteries. So these coronary arteries, coronary meaning heart, coronary is just all the arteries supplying the heart. And then cardiac veins, cardiac meaning heart, veins that are supplying the heart to remove deoxygenated blood from the heart. All right. So cardiac veins, coronary arteries, those are going to be the types of veins and arteries that we're talking about when it comes to the heart.